So I'd like to just begin by making, I guess, a single point here. This is really looking at the use of organoid cultures for cancer research by looking at the number of publications that report tumor or cancer and organoid in the title or abstract. And I think what you'll see is a dramatic increase in the use of these models. And I think it would be wise to expect that the numbers are going to continue to increase over the next few years. So the cancer research community is clearly embracing these models. I think that's fantastic. I think reflecting on some of the conversations we had earlier today about reproducibility and standards, I think this may also, give us cause for pause about maybe how we can support the community to use these models most informed way. Just as a little kind of bit of background, I think most people will be familiar with the concept of precision cancer medicine, and this is really the kind of key concept that we think about in cancer these days, where we're really tailoring therapy towards an individual patient based on some kind of molecular aspect of their disease, with the hope that we actually have a more effective treatment and we can reduce toxicity. And I've just illustrated a few examples here of some of the clinically approved precision medicines that we have for cancer today. And I want to make just two quick points. One is that you'll see that most of these work in relatively defined tissue types or histologies. And in most cases, they actually work in a subset of that tissue type, and often quite rare subsets of patients. So in many cases, as low as, say, for example, 2% of patients. And so this actually creates quite a challenge when you think about developing these types of treatments for patients. And the reality is today that actually it's about 10% of patients who really benefit from this type of precision cancer care. So just as a little bit of context, my lab has been using functional genomics approaches in cancer cells, and particularly cancer cell lines, for more than a decade. And I will get to organoids in a minute, but I do want to make a couple points about the cell lines themselves. And one is that we have a very large set of cancer cell lines in my laboratory, well over 1,000. And we have that to try and represent as much as this diversity that we know is represented in patient populations and impacts clinical responses. And over the last few years, we've very deeply genetically characterized these models, both in terms of functional readouts as well as genetic readouts, to use these in a number of different applications, from fundamental biology to target discovery. And these data sets are quite widely used. But a real key point for us is that we don't think this set is adequate. And we know that there's a number of limitations. So we know that many of these lines have been growing for some cases over 50 years. So apparently, actually, the HeLa line turned 65 this year, so it can officially retire. So we know that they have no genetic data that makes it germline genetic data or clinical data, which limits their usage. And we know that some disease types are particularly poorly represented. Of course, they don't really recapitulate well aspects of the tumor microenvironment. And a real key point for us is just that there's just not enough of them, because actually the disease is extremely heterogeneous. So there's big gaps. And so in fact, for many patient subgroups, we have no models at all. And so how can we really begin to move the needle on treatments for these patients when we don't have a system to study their disease? And for many of the approaches that my lab uses, which are quite high throughput, things in terms like biomarker discovery for therapeutic applications and target discovery, we need a sufficient sample size. It's not enough to see something in one cell line or one organoid, I think, to really have a robust finding that I think will clinically translate. Now, what's not clear is how many models we might want to have. And actually, it's quite a complicated question. I'm just going to walk you through one very simple way you could look at this. It's rather naive, but I think it illustrates the point. So we know that there's over 100 different types of cancer, according to the World Health Organization. Interestingly enough, there's not even agreement of this. Some organizations say there's over 200 types. But in the UK, almost all cancers occur at 24 sites in the body. And we know through extensive genetic sequencing of human cancers that many of the key molecular drivers of disease occur in this range of about 2 to 20% of human tissues, or human cancers. So you might say, well, actually, as a goal, as a community, to develop a model for any cancer that occurs where it has a driver that occurs at about a 2% frequency. So we have a representative model. And you could say, well, actually, if we did that, if you just said, well, how many models would I have to derive to achieve that? You might say, well, actually, I need about 50 models. And if you wanted 3 models to have some kind of statistical power, you might well need 150 models to actually have representation of these relatively rare genotypes, but actually that are still clinically relevant in terms of the way we think about developing treatments. And as I said, this is really naive. We actually know the picture is much more complicated than this. We know breast cancer, for example, can be divided into many subtypes. So I would argue that actually one of the most important applications of organoids, which is why we're here today, is actually to make a new set of disease models for cancer that much more accurately reflect the disease, that encompass this heterogeneity, and cover aspects like disease stage and ethnicity, which are largely absent from the current sets of models. And so to achieve this, we've established a network that we call the Cell Model Network UK to begin to derive more models of cancer. And this just shows schematically that network. So we have clinical sites across the UK who are partners in this, who identify and consent patients. The tissue is then taken from the patients through various surgical, usually surgery, and couriered to the Sanger Institute. We have about 10 couriers arriving per week, where we then derive an organoid from that sample. And we as well do an extensive genetic analysis of the organoid itself, as well as the primary tumor from which it was derived. And we're now making those models available through third-party repositories, such as ATCC. And actually, as of, I think, last week, you can now buy 28 different organoid cultures from ATCC, just like you would buy any other cancer cell line or cell model. And we're making the genetic data available through various data portals, and my lab then uses these for functional testing, things like drug sensitivity testing and CRISPR screening. So we've been working at this for close to 3 years now, and at the Sanger Institute, we have now processed 429 patient samples, so quite a lot. And this just shows our overall success rate across the 3 cancer types where we focused. So that's esophagus, colon, and pancreas. And overall, what we found is we have about a 40% success rate doing this. So we have an all-comers approach. We take anything that the surgeons will give us that they think is cancer, which creates some problems. But as you can see, based on this, we've now been able to derive 113 organoids. And for us, deriving an organoid culture that's a sustainable model requires us to be able to bank 25 cryovials and to do a freeze-thaw test of that organoid so we can grow it for 4 weeks after we thaw it. And if we're successful at that, then we call that a banked organoid. And our primary reasons for failure are numerous, but certainly poor sample quality is a big one, as well as low tumor cellularity in the sample itself. And this effort is part of a larger effort, an international effort that involves many other groups, including the Hubrecht Institute in the Netherlands and the National Cancer Institute, to really make a repository as a resource of new cancer models, including access to clinical data for those particular models, which we think will be extremely useful. So once you've made a biobank, this is extremely useful. And I'd just like to highlight very briefly one example where we've made an esophageal adenocarcinoma organoid biobank. And this was a cancer type where there was a real need for more models, because actually the cancer cell lines that were available were, let's say, of unknown provenance again, and also making mouse models for this disease is actually pretty difficult. So we made a very small bank, as illustrated in a few images here, and then we could leverage things like our large-scale drug screening platforms that we've used in cancer cell lines. And this just shows a small data set where we've taken 9 organoids and profiled their sensitivity to 24 different compounds. And what this illustrates is the more yellow, effectively, it means that the organoid is more sensitive to the drug. I won't go into a lot of detail, but I think there's some high-level things here. What you can hopefully see is there's actually quite a range of responses to these different cancer drugs. We can also see examples where organoids have very specific mutations or molecular changes that are predictive of response, and we recapitulate that in this data set. But I will say that, again, because we only had 9 organoids in this data set, it was very hard for us to make any kind of really robust statistical inferences about differences we see in drug sensitivity response. So it was rather anecdotal at this stage. I'm just going to skip the next 2 slides because of time. But just to say that obviously we're not the only ones that are doing this, and I've tried to summarize the majority of the papers that are available today where people have made tumor biobanks, often done very extensive molecular characterization of those and subjected them to drug testing. And these are some of the high-level, I think, consensus that has emerged from those papers. So overall, the organoids look like the tumors they came from, and I think that's broadly true. There are still some details there, but I think that's broadly true. In general, it doesn't seem that there's a huge bias in our ability to derive from different cancer types. This is critical if we want to have that representation of patient populations. But we also know that the cultures are often have multiple subclones that can evolve in culture over time. We're able to use these for drug testing and find the type of biomarkers that we know about. And I think importantly, we also see poorly understood variation in drug sensitivity. And this is where the opportunity is, because actually if we can explain this, this will be helpful for us. And in retrospective analyses, there is evidence that the organoids actually recapitulate the way that patients respond to drugs. I will say one note of caution on this: there have been no prospective studies that have shown how predictive organoids are in terms of clinical responses so far. I wanted to also just highlight something that I found very exciting in the last year is really the ability to derive autologous cultures or tissue slices that incorporate aspects of the tumor environment, in particular the immune system. On the right, actually, is effectively the paper that Calvin, as group, published this year, and he presented very briefly using this air-liquid interface culture system. On the left is a publication out of the Netherlands Cancer Institute by Emil Voist, where they've been able to derive tumor organoids and lymphocyte cultures from the same patient and generate long-term cultures and then mix them back together and look at aspects of, for example, tumor cell killing. And so I think this is a really important advancement, particularly in the setting of cancer, given the role of the immune system in cancer and the interest in developing molecules that modulate the immune system to fight the disease. So beyond the drug screening that I illustrated earlier, we have been developing CRISPR screening technology in the organoids. So we've recently published a study in 324 cancer cell lines where we did whole genome dropout screens to look for new oncology drug targets. And we think there is a huge opportunity to transplant this type of capability into these 3-dimensional organoid culture systems. And just shown here on the right is a proof of concept screen where we performed a genome-wide screen in an esophageal adenocarcinoma as a 3-dimensional culture. We were able to look for genes that were required for the fitness of that model. Some of the top hits were EGFR and ERBB2. We could subsequently take that culture, grow it in the lab, and treat it, in this case, with a small molecule inhibitor and recapitulate that result. So we think this is a huge opportunity. It's still quite challenging to do this and expensive, and so we're really working very hard now to find ways to make this much more efficient, and I think we're going to have a lot of success with that. I know my time is up, but I'm going to have 2 more slides because I think they will be of interest, and it's come up already today. I think there's a lot of interest in the setting of cancer to think about how we can use what are sometimes called functional diagnostic approaches to inform treatment of patients. or sometimes people talk about things like patient avatars. And the premise here is really taking the cancer cells out of the body, growing them ex vivo, testing them in some kind of ways, for example, their response to cancer drugs, and informing the care of the individual patient from which they were derived. We've seen really nice examples of that in cystic fibrosis. We've already talked about that today. And so, indeed, there may be an opportunity to do this in organoids. There is precedent here in hematological cancers. This has been done, albeit very rarely. And there are commercial vendors that will allow you now to grow your own PDX model and treat that mouse with therapy to inform your clinical care. But I did want to just kind of talk about some of the challenges that this brings. So to really achieve this, we really need safe and non-invasive ways to access tissue specimens. This is going to be deeply problematic if we don't have this, particularly as many of these patients might have poor performance status, meaning they're not very well. We know that heterogeneity is a problem. Sampling bias, do we have a sample that represents the disease, the bulk tumor? And we know that selection and culture are all possible confounders. We need ways of ensuring the quality of the specimen and its stability, and also overcoming some of the obstacles we face around variable culturing efficiency. We also need very good assays with very defined specificity and sensitivity that are really reproducible, scalable, and cost-effective. And I think it's still a little bit unclear about how this would be really compatible with clinical timelines. When would you really use this that you could get a result fast enough that you could inform clinical care? So I think, I don't want to be overly cynical here. I think there is an opportunity. I think these are all manageable challenges, but there are a lot of important things that we need to do before this, I think, will really be implemented in the clinic. And with that, I'd just like to thank the many people involved. Thank you.